Okay. So welcome everyone to the second online MOOC for learning AMR and screening in theory. And this is presented to you by Jogle, the Microbiology Society, Kamasi Hive, and MUST as well. And we're going to be learning about the techniques involved in identification of microbes. So just having a little recap, these MOOCs are all hosted on Jogle website on Challenge AMR MOOC Africa. You can just search for MOOC in AMR. And today is the 28th of September, and we're learning about identification. Last week, we learned more about microbiology, bacteriology 101, and how antimicrobial resistance emerges. We heard from myself and Courage Sava as well. And also we shared a link for a certificate of attendance. So at the end, you can get a certificate as well. And then next week, we'll be learning about sensitivity testing, antimicrobial susceptibility, then AMR, digital mapping, and we'll be learning about Harry's app. Harry's with us as well, he's a panelist. And then later on, we'll have practical sessions as well. So based on your attendance, we'll be inviting people to join Linda and Harry in Ghana and Tanzania on their work as well. This is the team. We've got Courage, we've got Linda, Gamli, Joy, Harry, myself, and John Moat, who will be speaking next week, and perhaps more, we'll see. So today, to introduce to you Dr. Linda Salekwa, and we'll be talking about techniques involved in identification of microbes. Hi.
Thank you, Chris. Hi, everyone. As Chris said, I'm Dr. Linda Salekwa. I'm a lecturer at Mbeya University of Science and Technology here in Tanzania. And today I want to discuss with you a bit on techniques which are involved in identification of microbes. I hope you will enjoy the session. To begin with, we should all understand that we can isolate microbes from different parts. We can isolate the microbes from air, from water sources, we can get from the soil, and even from the sewage systems. All those parts can act as sources for microbes. So depending on your purpose, depending on what you are looking for, depending on your interest, you can either acquire your samples from what I've just explained and try to isolate the microbes. And depending on the technique and depending on the purpose of your study, you can now select on which technique or which method I can use to select my microbes. Then we can learn on different techniques. We have microscopy, on which we use microscopes. We have, which is direct examination. We have cell culture. We have serological assays. We have molecular assays. All this depends on your interest. Depending on what you are looking for, you can select which techniques applies to. Then when we start with microscopy, for a case of bacteria, we can use the microscopes, especially light microscopes, to observe or to check on the morphological structure of your bacteria. And you can also observe the colonies which are grown in this culture and get to know the shapes which is presented by the colony, the color of the grown colonies, and the smell which is given out from the culture. So this is the first style when you're doing microscopy, and not that microscopy, when you're doing microscopy for bacteria, you have to use light microscopy. But now there are cases that you have too much sample, like then you cannot just go around and test the whole of your thousands mils, for example. So what you have to do is you use filters, which has the size smaller than the size of your microbes of interest, and you can filter the samples to obtain the filtrates. Then you can take them either to the microscope and observe what you see, or you can take them for culture. so that you can see what grows out of your filter. So this is an example when you have a filter which is used to filter a sample, maybe it was a water sample or milk sample or any liquid sample. You can get those ones and culture. If you can see in B, you can see in B, you can see it is grown. The culture is put in media and allow the microbes to grow. So you can observe the colonies which are grown from the culture. Now, another technique, that was microscopy. Now, another technique is culture. Sometimes you may be interested in getting live microbes. And for this case, you have to grow your bacteria. And there are different kinds of culture that can be used. Depending on the state, you have the solid, we have liquid and semi-solid. For the solid, we have a number of them. We have, for example, maconche, chocolate, nutrient, agar, blood, agar, depending on the contents of the media. Also, we have liquid media, and we have, which we term as broth, which is also used to grow some bacteria. Another one, we have semi-solid. This is used to, this is mostly used when you are studying the motility of bacteria species. And for the case, you can just appreciate, you can see the inoculated tube, you can see the clear media. But when you have a negative motility bacteria, you can just observe that there is just a line, a single line, which shows the growth of microbes. But when you go to the positive test, you can see that the color has spread in the whole media, that you can see that your bacteria is motile. So semi-solid media is mostly used to observe bacterial motility. As I said, there are different types of media that can be used to grow bacteria. And this depends on the composition. When you consider the composition, we have 4 different types, where we have simple media, where we have complex media, synthetic media, and special media. All these are determined by the purpose of your study or the bacteria strain that you are looking for. If it's a fastidious bacteria or a non-fastidious bacteria, will depend now on what should I put in. Another one, it depends on the roles you want your media to do. It depends. If you want to differentiate, you can use differential media. There are elective media, there are selective media, and enrichment media. Well, this, depending on the roles that you want that media to help you, that is how you can determine your media of interest. Now you have your cultures, they are grown already. You can identify the kind of a bacteria inside that culture depending on morphological characteristics, as I said earlier. This can be by observing the colonies, but you can go further and stain your bacteria by using gram staining or other staining, as I'll explain later. Or you can use the biochemical tests, which can easily differentiate between the similarly looking structures. But also we have serological assays, we have molecular assays. All these can help you to identify the strain of your bacteria in the culture. Coming to the morphological identification for the case of bacteria, we know we have different shapes of bacteria. We have bacillus, we have cocci, we have spirochete, we have coma, we have cocci, depending on the strain of bacteria. So now, knowing or understanding that you have various shapes of bacteria, you can now know which if you are bacteria of interest from which shape, then you can observe and know that this bacteria I'm looking for is supposed to be in a coma structure. So if you see something different, that you know that is not what I'm looking for. This is for the case of morphological. But as I said, again, you can observe the size of the colonies. You can observe the shapes of the colonies. You can see the profile, whether it's concave or convex, or flat. You can identify the margins of your colonies. You can see the surface, if it's a rough surface or if it's smooth. You can see the transparency of the cultures. You can also identify by colors. You know that the certain bacteria gives this color of colonies when grown in which media. These characteristics normally depends on the media you are looking for. I mean, the media you're using to grow your bacteria. But also you can see the consistency of the colonies or the smell which is given by them. So with these characteristics, you'll be able to identify which colony depends to which bacteria. Another thing is when we do gram staining. Gram staining, this is the most popular staining, which is used to differentiate between two groups of bacteria, gram positive and gram negative bacteria. As discussed in MOOC 1, Chris tried to explain very well about the structures of the cell wall, which is the one differentiate which to be gram positive and which to be gram negative. Here you can see the size of peptidoglycan layer. You can appreciate in gram positive that it's thicker than in gram negative bacteria. So what we expect is that when you do the primary stain, we expect that those with from the gram positive group, they will retain the primary stain. And then when you go for the gram negative, since the peptidoglycan is very thick, it won't be able to retain the color when you treat them with any colorizer. So now when you apply the secondary staining, the gram positive bacteria will pick up again the blue color and make it purple. So the gram positive bacteria will be visualized as purple in color. But for the gram negative bacteria, since after the colorization stage, the wall of the primary stain have been washed away, now the gram staining bacteria will stain the color of the counter stain. That is when you will observe the red colors. So that is easily how we differentiate between the two. And this is how you will see them. You will see clearly the gram positive staining purple and the gram negative staining red. Now, there are cases that we have gram indeterminate bacteria. These are bacteria which cannot be differentiated by the gram staining. This is due to the different composition of the cell wall. For example, we have acid-fast bacteria. These bacteria, they have another thing which is known as microbes acid in their cell wall, which makes them not very good in responding about the two staining, which are used in gram staining. So for this case, this bacteria contain the microbes acid. For example, we have Mycobacterium species. They are staining by use carbofascin, which is used to differentiate between the acid-fast positive and acid-fast negative bacteria. So this is for the case of those who contain mycolic acid. But we have also other bacteria which, when you stain, when you stain with gram positive, with the gram stain, you find out at some cases they tend to catch up all of them. Some colonies are seen red and some are seen purple. So when you see that, you can easily know that this bacteria can be differentiated by using gram stain. So structure of those with mycolic acid, you can just see here the mycolic acid, which is in this bacterium species, and this is the one which makes them not respond very well with gram stain. Another one, we have biochemical tests. These are tests which are used to differentiate between different colonies of bacteria or different strain of bacteria with or without a certain enzymes. So we have this, for example, we have urease test. This is special for those organisms with the ability to split urea, and then it causes the change of the media to red. And an example, we have Klebsiella and Helicobacter. These are urease positive species of bacteria. So whenever you have the urease, whenever you have your media and you find it when negative, it stays like this. And after growing your species, it changes to pink. You can know that the bacteria that have grown in this culture is urease positive. It has that enzyme which is capable of splitting urea. It has urease, which is capable of splitting urea. Another one, we have indole test. This is another test, another biochemical test, which is used to differentiate those bacteria with the ability to decompose amino acid tryptophan and give the indole, which tend to form a red, a pink ring on top of the media. So you can see here for indole positive, you see here, you see a ring, while in negative it's not red. So you can know that if you see this, you can know that your bacteria is capable of degrading tryptophan. Another one, we have catalase, which is this one differentiate those organisms with ability to convert hydrogen peroxide into water and oxygen. And for this case, you will see bubbles in your media. This one will show that the oxygen has been formed. And since your media contains hydrogen peroxide, it means that the bacteria you have grown has the ability, has the enzyme, which can degrade the hydrogen peroxide inside the media to give water and oxygen. That's why you can appreciate by the bubbles. Then Pseudomonas is an example of the catalase positive bacteria. We have another one, which is nitrate reductase test. This also tests those organisms with the ability to reduce nitrate to nitrite. So you can see here, when you have reduction to nitrite, you can see the change in color. But when you don't have reduction, you see the gray colors. Then we have the API tests. This one contains a group of biochemical tests in one test, which can be used to differentiate a certain group of bacteria. For example, we have API 20E. This is one of the examples of API tests which are used to differentiate Enterobacteriaceae, a family of Enterobacteriaceae, by using this API kit. And it helps you to know which strain of the Enterobacteriaceae is in your media. So here you can see we have 10 biochemical tests, and on the other hand, we have 10. So all of them, it makes 20 biochemical tests. So with these tests, when now you have the kit, and you inoculate your samples in all of the 20 tubes, and you can incubate for 24 hours. After some hours, some of the tests require addition of a certain reagent and some doesn't require. But when you add reagent in the respective wells or respective well of the kit, you can observe the color change. And when you see the color change, you can know that your bacteria has been grown and it is positive to this biochemical test. So at the end of the day, you can take the results and take back to the chart. There is a chart which will help you to identify which is positive and which is negative. So you take this one and go to the chart and see which one matches what. So when you see pink, for example, in this, for example, you see pink in urease, you know that it is positive. So you just go and have a chart and look which one corresponds to what. Then you can come out and say that this bacteria which is positive to, for example, to catalase, urease, and this one is E. coli, for example. So that one can help you to identify which strain of bacteria inside the sample. So this is one of the tests, or one of the kits which has been used to differentiate different strains of bacteria. Okay, there are some cases that you just need to identify a certain strain of bacteria in your samples, but there are cases that you need to quantify the number of bacteria. For example, in a setting, there are some cases that the presence of a bacteria does not matter, but what matters is the number of bacteria in the sample. So there is a need to have a quantity, or to know the quantity of bacteria inside your sample. And most of the time, we measure them using colony forming units. And how we do it is that you take your original sample and dilute it to a level that you can easily count without any problem. So for example, here you have the original inoculant, and from the original inoculant, you have to take 1 ml and you mix with 9 mils of your diluent, and then you plate. When you plate, you can observe that there are 2 numerous colonies that you cannot even differentiate. Then you can go to the second dilution when you take 1 ml from there, 1 to 10 dilution, and then you have now 1 to 100 dilution. You can see when you grow it, it's a bit clear, but you cannot count the number of colonies. You can just dilute it further to 1 to 1,000 dilution, where you will have clearer colonies, but still you cannot count from this dilution. So you dilute further to the last dilution that you feel very comfortable to count, and then you take the mean, because you do it in duplicates or in replicates, so that you can not rely on only one attempt. So you have like 3, 4, and then you can take a mean count from the dilution that you feel it's okay for you to count. Then when you count them, you times with the number of dilution, and then you can get the number of bacteria that you're expecting in 1 ml. So at the end of the day, you will count the colonies and get the number of colony in the colon forming unit per ml. So from there, you can now report that my sample had this strain of bacteria at this amount. So this is one way. Most probable number is the one way that is used to enumerate the number. Now we go to viruses, but due to their very tiny size, we tend to use electron microscopes when we are checking on the morphology of the viruses. And we have like 4 structures, the common structures of viruses. We have the spherical, for example, the influenza virus. We have the helical, for example, tobacco mosaic viruses. We have adenovirus, which is polyhedral. And we have the complex one, like bacteriophages. So these are some of the structures of the viruses which are common. And you can just observe by using the electron microscope. But it's not very easy to visualize viruses. Coming to the culture of viruses, here we have a different case. Unlike bacteria, we use the media to grow them. But for the same of cultures, as we all know that a virus is normally dead when outside a living organism. So in order to make it alive, you must have a live cell or a live organism. So there are 3 ways that you can use to grow viruses. We have living animals like lab animals, mice, rats, rabbits, any animal that you are deciding to use to grow your viruses of interest. But we also have embryonated chicken eggs. This also can be used to grow viruses. We have cells and tissue culture. So depending on the part of an organism which supports the growth of the virus, you can just go further. For example, if the virus tends to attack the liver cells, you can go and isolate the cells and grow them in culture, and you can use the cells to isolate the virus of interest. So we have cells and tissue culture, we have the animals, live animals, and embryonated chickens. These are the ways that these are commonly used to grow viruses. Then coming to the virus identification, it's also different. Because in viruses, we don't have colonies. So for the viruses, what we tend to do is to observe the effects brought by the virus growth in a cells or in an organism which is to a virus. So if, for instance, you have used the cell lines, you observe the cell line, the characteristics of cell line before you grow your virus. And then after you grow your viruses, you can observe the differences. For example, what you see in front is these are Vero E6 cell lines, which are used to grow a number of viruses. But for this case, we are using to grow Rift Valley fever virus. These cells, if you see in an infected part, you can observe a mesh-like structure where you can see the intact structure of cells, which is called the confluent monolayer. We don't see any space in an infected cell. But when now you infect and allow to grow for 5 hours, this is what you see. You start seeing some of the structures starting to grow in your cells. But when you allow now for 8 hours, when you observe, you'll see the wall structures of your cells has been distorted, and you can now see the spherical shapes. But when it goes, you will find that the whole cell, this monolayer has been worn out by the growth. So when you see this, the effects that you see in a confluent monolayer, these are the so-called cytopathic effects, including the plaques, including the change of shapes of the cells. These are the ones which can help you to know that your virus of interest has been grown. So from this one, you can just know that since this cell line supports the growth of these viruses, then this is how you can select which cell lines to be used. Because you may find out that you grow a virus which is not susceptible to the cell lines, so you find out that the virus is not growing because that the cell lines does not support or do not support the growth of your virus of interest. Now, as we said in bacteria, there are some cases now we want to quantify your viruses. As I said, the species you see in cell lines is the so-called plaques. So the number of plaques shows you the number of viruses. That's why when you take from the inocular and grow it directly in your cell lines, you will find the number of plaques too many to count. You cannot even quantify. But when you dilute it again, you can see that the number of plaques are reduced. For example, here, if you have a virus stock and you take for 0.1 ml and you grow it at a 10 dilution to 100 dilution, 1000 dilution, you can find that the more you grow them, the more the plaques are reduced. So you reduce the dilution till to the point that you can clearly count the number of colonies in the sample. Then after getting the number of colonies, then you can find the average number of plaques per ml. Then you can get the plaque forming unit per ml. So you can now see for bacteria we call colon forming unit because it forms colonies. And for viruses, you see plaque forming unit because it forms plaques. So the number of plaques will help you to quantify the virus. And this is very important, especially in vaccine studies. When you are doing the dosing of vaccine, you have to quantify the number of particles that you need to inoculate in an animal to give a required immunological response. So you have to take your your vaccine dilution and quantify the number of particles in order to get the required dose of the virus particles. So plaque assay is one method, but we have another one, which is TCID 50. This is also, you take the inoculum and you dilute it and get the different number of dilution. And from there, you can plot in a graph and get a number of viruses that you are expecting from the virus. Next, please. This is the TCID 50, a tissue culture infectious dose. This is also, as you can see, you have the serum dilution. Here you dilute. Now here is a different case. You have the viruses, and then you have serum dilution. So now you can, the serum tends to, we understand that the serum will contain antibodies against that virus of interest. So when you inoculate a serum with a plaque, you expect the antibodies in the serum to neutralize the plaques in the samples. Next. Now, if you remember at the beginning, I just explained that what determines the type of assay to be used is the interest of a researcher or the interest of a person. So if you are interested in the whole organism, if you're interested in the colony, it means that you have to go forward and do culture. But in some cases, you may not be interested in getting the culture, or you may not be interested in getting the whole organism. You're just on the antigenic part of the organism or the antibodies also against that organism. So this one, we have the serological assays. We have a number of them, but the most popular one, we have enzyme-linked immunosorbent assays known as ELISA. We have hemagglutination assays, we have neutralization assays. All these are known as serological assays, which tends to base on the issues of antigen and antibodies detection. So depending on your interest, you can decide if your interest is the antigen, then you will go to use the antigen ELISA. If your interest is antibodies, you can go ahead and decide which type of ELISA you want to use. But also, if you want to observe the antigen, antibodies neutralizing antigen, you can just see the neutralization assays and the hemagglutination assays, and you can see the reaction which occurs between the antigen of interest and antibodies of interest. So for example, if you are just trying to check if an organism, make example, a human being has antibodies against, let's say, E. coli, it means that you have your plates with antigen, then you take the human sample, a positive human sample, which you know that it has antibodies against that E. coli. So when you mix up in an ELISA plate or in an immunization plate, you can just observe that reaction between antigen and antibodies. So depending on the type of assays, you can appreciate what and if the antibodies or the antigen of an organism of interest are present in it. But you can find out that an organism displays different types of antigen. So you find like it's the same species of organism, but this species contain a certain antigen in the wall, while the other one contains a different antigen. So what happens is that you find out it's from a single strain, from a single single species, but you find out that it has different antigenic structures. So you'll find out that you'll get from a similar strain, you get different antigenic presentations. So from this, we can work and get the so-called serotypes. So the serotyping is one of the techniques which is used to group different organisms from a certain strain. For example, here we have Salmonella typhimurium. It has some of them they display O antigen, while others display H antigen. So you can get from just the two antigen, you can get two different serotypes from the similar species of Salmonella typhimurium. Next, if you do if you go ahead and try to map using the informatics tools, you can find that a certain group of organisms which has the same antigenic presentation falls into a certain group. For example, here you can see there are groups of organisms. These are the so-called serotypes. They share a similar antigenic structure and present a similar anticell. So you can see here you have different groups from the similar strain of bacteria or virus. But now you may be interested only in the molecular part of the organism. And here we use the molecular assays. We have a number of them depending on the molecular content of that organism. You find that some of organisms have the deoxyribonucleic acid, while others have the ribonucleic acid, the DNA and RNA. So depending on which type your interest is, you can determine which type of molecular assays I'm going to use. For example, we have the normal PCR, which is used to amplify the DNA content, but we have the reverse transcription PCR, which is used to amplify the RNA content. It means that you have first to convert the RNA into a DNA before you allow amplification in a polymerase chain reaction. But then getting the amplicons, after getting the amplicons, you can go further and do the sequencing to get the nucleotide sequence of your interest, and then you can we can use metagenomics to observe a huge or the whole part or a bigger number of organisms from the environment. So now, as I said, for example, in bacteria, we expect to get the plasmids and the extra chromosomal DNA. And from the viruses, depending on the type of virus, if it's a DNA virus like a bacteriophage, you can go ahead and get a DNA. But if it's an RNA, it means a tobacco mosaic virus, you have to go ahead and get an RNA. So a technique which is used to isolate a DNA or an RNA from the bacteria or from the virus is known as it's not a DNA or RNA extraction. So you go ahead and do the lysis of the cell wall and you obtain the DNA, and then you can have your DNA, which is a mixture of everything that is in that organism. So if you just obtain a DNA from a human sample, it means that it contains the DNA of that human and all the microorganisms that are found in that human sample. So you'll find that when you do the DNA and RNA extraction, you find you have a mixture of DNA, including those that you are not interested in. So the way that you can differentiate the one which you are interested in is just to go ahead and amplify only that DNA of your interest. So when you go to amplify it, you just do it by a primer. This is a short oligonucleotide sequence, which is used to initiate amplification of a DNA of interest. So let's say you have a mixture of 1,000 of DNA samples from different sources. Now you just want to amplify that of interest. So from there, you go ahead and design a sequence which will initiate amplification of only the DNA of interest. So you allow the first step of a DNA of PCR, which is denaturation. Since we know that many of the DNAs are in double strand, so you go ahead and do the denaturation part in order to separate the two strands, in order to get the one strand and the other. So when you get the single strand, you can go ahead and do the annealing. This is a bit lower than the denaturation. Normally the denaturation tends to be above 90 degrees. Then you can come down to around 50 to 60, which is the annealing temperature. And here you allow now the DNA, the primer which you have designed specific to a certain DNA to bind to the DNA of interest. So when it binds there, it means that it initiates amplification of the DNA of interest only. So that's why it's very key to know how to design your primer so that it is as specific as possible. So when you allow the annealing temperature, it means that you have allowed your primers of interest to bind to the strands of interest. And now you can go to the polymerizing stage, where you allow now the extent of the primers, copying the template strand. So at the end of the PCR reaction, you'll find that you have a double of each of the template strand that was amplified. So when you're doing the designing, you have your strand, which normally runs from 5 to 3 and 3 to 5. And from this one, you design both the forward primer and the reverse primer, because you know it has to copy from 5N to 3N. So it has to be, one should run forward and the other one should run reverse. So at the end of the day, you get the whole thing copied. Then after getting the primers of interest, it means that you can go ahead and amplify your sequence of interest from a pool of extracted DNA or RNA samples. So we have different types of PCR. We have the multiplex. Here we use more than one pair of primer. As you see, the primer works in pair. You have a reverse and a forward, but this belongs to one. It's a one pair. But there are cases that you can amplify a number of strains by using various pairs of primers. So when you are doing a single reaction with more than one primer, it means that you are doing a multiplex PCR. But now we have a nested PCR. Nested PCR is more sensitive. This is because you just run your first round of the PCR, and then when you get the amplicon or the end product, you just go ahead and take the end product as the samples for the second round. So it's like a chain of PCR reaction. So this one tends to increase the sensitivity, because you can just go ahead, for example, for the first round, you put primers, which are not very specific, just to narrow down the amount of DNA that are amplified. Then for the second round, you use primers, which are more specific. For example, if you have a mixture of samples, maybe you have DNAs from viruses, from bacteria, from protozoa, you can use the first round just to amplify DNA samples from bacteria. It means that at the end of the PCR reaction, the sample will have too much of the bacteria DNA because those are the ones which have been amplified. Then from there, you can go ahead now and use primers which are specific to a certain strain of bacteria. So it means that the first round, you have just reduced and omitted all the DNAs from the viruses and others, and remaining high number of DNA from bacteria. Now you have a high number of DNA from bacteria. You can go further and use specific primer and amplify specific strain of bacteria from your pool of DNA. So that is the next step. You have another one, a reverse transcription. As I said, this is used when you have you are amplifying an RNA sample, because you have to change it to a DNA, that is reverse transcription, and then you do the polymerase chain reaction to amplify the DNA of interest. Next. Now, since you have the DNAs, I mean, you have different microbes from the microbial community, you can go ahead and extract the DNA from the community. When you extract the DNA, you can go and do the PCR, where you will amplify only the sequences of interest. And this one, from the sequence of interest which have been amplified, you can go for sequencing and see the nucleic acid, I mean the nucleotide inside that DNA which have been amplified. And that is amplicon sequence. But then there are some cases that you don't go further and have the primers. You just sequence whatever is inside your sample. And that is metagenomics. But when you do the metagenomic sequencing, here you have to be very good in analysis of data, because you get too much DNA, I mean too much nucleotides, and you may not be very good in analysis. So it's very good when you are targeting new species, because you don't have any reference to help you in designing the primers. So when you do the metagenomics, you have a bigger chance of getting new species, and but you must be very good in analyzing the results. You must be very good in the informatics skills. So this here you can get a summary that you have the microbes. Then you can go ahead and get the metagenomic sequence. And then when you get the metagenomic DNAs, you can go ahead and check the nucleotide sequences from the DNAs that you have isolated. Depending on your interest, you can do whatever you want to do. For example, you can do in silico analysis and get what you can see there. You can get the taxonomic assignment. You can get the phenotypic, I mean phylogeneticity predictions. You can do the genomic reconstruction, depending on your interest. But you have the databases, different databases in the NCBI, for example. Here you have different databases, reference databases, depending on your interest. So now having those data, the nucleotide sequence data, depending on your ability to work with them, you can go ahead to the gene, the point of gene level, and identify the genes inside your DNA. So it's depending on what you want to do. You can just go ahead and see the messenger RNA. You can go ahead and get the protein sequence. You can go ahead and get the amino acids and the nucleotide sequence. So you can get as far as possible, depending on your knowledge in bioinformatics, analysis of the DNA, of the nucleotide sequences. So for the case of our issue now of resistance, it means that now we are going to the issue of antibiotic resistance. What code for antibiotic resistance is genes for antibiotic resistance. So if an organism is expressing resistance against a certain antibiotic, it means that it has already developed a gene against that. So if you go down to the molecular level, you will find the genes which code for that antibiotic. And it's good enough that we have now the CAD. This is a comprehensive antibiotic receptor database, which consists of different sequences from different resistance genes which has been isolated so far. So whenever you get your gene of interest, you can go further and blast, for example, in this CAD sequence and get which gene is coded by which antibiotic resistance is coded by that gene. So the CAD, it contains the sequences of a certain antibiotic resistance which have been already isolated, and then they are kept in that database. And so it enables you to know a specific strain, a specific antibiotic resistance, which is expressed by certain organisms. So from that one, we got antibiotic resistant. These are the genes, a collection of genes which codes for a certain antibiotic resistance. So we can now appreciate that since bacteria or microorganisms tends to exchange the genomic. I remember in the previous MOOC, Chris talked about it, on how microorganisms can exchange genetic makeup between themselves or from the environment. So it means that they are capable of now transmitting the gene coding for certain resistance from one bacteria to another, or they are capable of taking it from the environment. So it means that when the environment contains certain antibiotic genes, it means that bacteria can take it and now express it and transmit it from one species to another. So you can see here from different human activities which can create antibiotic resistance, which can facilitate antibiotic resistance. For instance, now that we are having the COVID pandemic, it means that people are using a lot of antibiotics. It means that we can predict in the coming years that we are going to have a very serious antibiotic resistance problem, because people are now using, when someone gets maybe some symptoms that looks like COVID, they just go ahead and take antibiotics just to rescue themselves. So you will find yourself that at the end of the day, we find ourselves that we have used too much antibiotics, we have used them in a wrong way, and we have developed too much of antibiotic resistance. And we can now, we are all witnesses that the types of antibiotics which are used are those strong antibiotics. So you can predict what will happen in the next few years due to this pandemic that we are facing now. So we also have the farming systems and the animal husbandry. Some of the agricultural experts, they are using too much antibiotics products and even into animals and treatment of their animals, which makes which helps or which facilitates the formation of antibiotic resistance. So if now we compile all these activities, we compile the issues of husband, we compile the issue of we using the antibiotic resistance. I may not know what happens in countries, but as far as my country is concerned, people are not very serious with how we use antibiotics. So we still have a long way to go in educating our societies or in educating our people on the right way of using antibiotics, the antibiotic stewardships. It means that we need to educate them in order that we minimize the contribution, our contribution in making this antibiotic resistance problem exacerbate. So we find out that those from the human activities and from our activities and from the animals' activities, the sewaging systems, the food products, the travel, you find out that too much of the antibiotics are accumulated in soil, in water bodies, in wild animals. And from there, we are just making the sources of antibiotic resistance. So bacteria in there are exposed to a number of antibiotics, and then they tend to develop resistance in the natural environment. Now, provided that they are in the environment, it means that we are no longer safe. It will just come back to us from one way to another. So we can just see how big the problem is. If you have appreciated what Professor Courage Kosi has just presented in the previous book, in book 1, you can just appreciate what is happening in most of our African countries due to the lack of proper use of antibiotics. And so I can make a summary of what we have discussed in my presentation. That we have discussed the isolation of strain from the food samples, or from the water samples, or from the soil samples, or from the air, whatever that you want to get your samples from. Then we discussed in the microscopy of both, microscopy of bacteria and microscopy of viruses. We have appreciated different morphological structures of viruses and bacteria. We have also seen the use of filtration method when you are trying to check the microbial content in huge samples. And we've seen the culture medium, and colony features. We have just seen the different staining methods like the gram staining and acid-fast staining, and then the biochemical tests on how the biochemical tests can be used to identify the strain. We have the API test, which is a collection of biochemical tests. We have the bacteria and virus quantification for different reasons, as I said, specifically or especially for the viruses dosage, when you are calculating the virus dosage, or when you are calculating the bacteria content in your samples. We have also discussed serological assays on how you can determine the antigenic parts and antibodies against a certain microorganism, and molecular assays, basically PCR. This is when you talk of molecular, the PCR is the mother of all. And then the use of sequencing in helping you to interpret what is inside, what is amplified in the PCR. Then this marks the end of my training. I hope you have enjoyed. If you have any question, you just go ahead and ask. Thank you very much.
Okay, thank you, Linda. That was amazing as always. I'm going to stop sharing the screen so that we can see the participants.
Yes,
Questions? Okay, I'm gonna make it so. Okay, there's one attendee with their hand raised, and then I'll just allow them to talk. And there's a few questions as well. So I think you can talk now, Raquel. Can you talk? Okay, in that case, I'll just go to the Q&A questions that we have for you. So Rita says, hi, good morning. What biochemical tests can I use to differentiate Klebsiella isolates to subspecies? Just see if we can find these people to ask them as well. I'm allowing Rita to talk. And the other question askers to talk as well. So you can ask it in your own way. Hi, Linda, did you hear what I said? Sorry.
Hello.
Linda?
Yes, good morning, everyone.
Morning.
Yes, so I wanted to ask which type of microscopes would mostly be recommended for wet prep and stain slide examination, please. Thank you.
Okay, it seems like Linda might have some technical issues. But I'm also a biochemist and do these kind of things. So I think just a regular light microscope would work for it depends on the level of detail. To be honest, you want between 60 and 100 times zoom. You don't need any confocal or anything fancy like that. But if you're able to obtain 60 to 100 times zoom, you should be able to see the shape of the cells, certainly. And if it's a light microscope and you've used gram staining, you should be able to see the color as well. If you're looking for fluorescence or for higher intermolecular, interbacterial detail, you might want to use a confocal or something like that. but a regular one should be fine, or a phase contrast even should be fine. Yeah, so then 60 and 100 times zoom should give you the ability to see most bacteria. Yep.
Okay, thank you very much.
If anyone else would like to ask a question as well, I might be able to answer for Linda while she's having technical issues. I think Rita and Hoja can both talk, and so can Akela, if you'd like. Okay, I'll just look in the Q&A and answer those questions instead. So we have, how could one isolate metagenomic DNA from microbes? So you can just get it from the environment. You can also get it from microbes. So if you have isolated a microbe, you might have found in the environment, taken a sample. You've then reduced the count of bacteria by serially diluting your bacteria, for example. And then after you've serially diluted the bacteria, you have isolated a particular colony. Maybe you've done some identification. You might have looked through a microscope. You don't need to do that. But you can do that if you'd like. You can then grow up these bacteria, or you can just take straight from the sample itself, and you can use a set of random primers. So there's lots of primers that are sort of generically bind in different places, and they can give you amplifications of various parts of the genome, or you can have more specific amplification as well. And you just add sort of a lysis mixture to the cells to allow the cells to get rid of the cell walls and let the cell membranes break apart. And then it's basically you do a genome prep, and they sell it in kind of kits. And then with this genomic prep kit, you can then isolate just the DNA itself. And then from that, you can do a PCR using the generic primers that I talked about before, all the specific primers. And then you can also do this a few different types of metagenomics. You can do quantitative, or you can do non-quantitative. If you do quantitative, you can do a certain set of, you can do a certain number of PCR cycles, or you can do even as many PCR cycles as you'd like, perhaps even 50 plus, 80 plus, to just use up all of the reagents and see what quantity of fluorescence you have, and you can therefore see the different genes and how much of that gene there is. There's a few different ways you can do metagenomics, but the simple method is find the bacteria or your sample, lyse it to make it so the DNA is available, and then do your big PCR set, and then sequencing from that PCR set. Yeah, I hope that answers that question. Is Linda, are you back? Linda?
Yes, Chris.
Oh, you're back. Okay, great.
Sorry,
my
internet is a bit problem.
Okay. Do you think you'll be able to answer me?
I can answer some questions in the chat.
Okay. You'd like
if you can't hear me correctly, I can read from here and answer.
I think we can hear you.
Yes, Chris.
Yeah,
can you hear me?
Yeah, we can hear you.
Hey, can you hear me, Chris?
Yeah, we can see you as well.
I can't hear you.
Okay. So Linda can't hear us, but we can hear her. Sorry about this. Okay, so whilst Linda's figuring that out, I'll see if there's any more questions. So after dilution, samples are inoculated on plate count agar. I would like to know if it's possible to skip this method and go directly to the biochemical test. So I see. So after dilution, so in this case, Monia, are you suggesting that you can take it straight from the sample without isolating your bacteria? I think, as Linda says, the test will work, but you might have an array of bacteria rather than just the one particular bacteria. So it depends on what the use is of the assay, I suppose. If you're looking to see if there is that certain type of bacteria, then perhaps it could be a way to do it, but it doesn't rule out the fact there are other bacterial species in the sample. What best way can we archive resistance genes, microbes, and what temperature for further research? So there are an array of different sites where you can find genomes of bacteria, and you can actually annotate them yourself in sort of citizen science kind of research projects. At what temperature for further research? I'm not sure. But we're doing lots of DIY next week, and so in terms of temperature, there are a range of temperatures that you can use, but actually you can not only use incubators which are 37, you can also just do it on the bench side as well. So next week, John Moat, who is part of the AMR screening facility at Warwick, he does a lot of his work at the bench side if it's cheaper, but also using the 37 or the 30. 36 is actually the perfect temperature for most bacteria. The storage of bacteria, normally you would be expected to use -80, and most people do. However, long-term storage, and that's for long-term storage up to 10 years, and you can even freeze dry your bacteria. But in fact, -20 is fine as long as they have cryogenic freezing agents with them, cryoprotectants, for example, glycerol, or even skimmed milk is fine, as long as it's sterile, of course. And then if you resurrect these bacteria from these glycerol stocks at -20, it works almost just as well, but it doesn't last 10 years. It's more like 1 to 2 years before you start seeing contamination or loss in life. Yep. Any more questions? No, I think that's it. Thanks a lot, everyone. I'm going to stop the recording now.